Good morning dear students of class 8, and I am happy to welcome you all to today's zoology class. What are we going to learn today? Today's topic is about blood groups. So what are we going to learn today about? Blood groups. You all have a blood group, right? If you don't know, just ask your parents what your blood group is. So you may have blood groups like A blood group, B blood group, AB blood group or O blood group, right? So each one of you have different blood groups. We all are human beings, but we have different types of blood groups. What is different in everybody's blood? Why you call it by different names like A, B, O, all? About this we'll be learning in today's class. So let's get started. So before learning about the types of blood groups, let's learn two important terms. What are those two important terms? One is going to be the word called antigen, and the next word is going to be a word called antibodies. So first let's learn about antigens and antibodies. What is an antigen? What is an antigen? Antigens are proteins, just like the enzymes are proteins, right? Just like the enzymes, antibodies are also proteins. Okay, where are they present? Where is antigen present in your body? Antigens are present on the surface of red blood cells. These are red blood cells, no? So you have studied already, red blood cells are biconcave disc-like structures which give red color to the blood. So on the surface of the red blood cells only antigens are present. These antigens are of two types called antigen A and antigen B. What are the two types of antigens? Antigen A and antigen B. See here, this is antigen A and this is antigen B. What difference can you observe in both of them? In A, you can see the antigen as small ball like structure, and in antigen B you can see spike like structure or a diamond shaped structure fixed on the red blood cells. Clear? So this is the difference, that is they differ in their shapes. So antigens differ in shape and they are present on the surface of red blood cells. This is about antigens. What about antibodies? Antibodies are again proteins. Antibodies are also proteins. Where can you find antibodies? You can find antibodies in the plasma. What is plasma? Have you heard this word? We have already studied in this lesson, no? In the earlier part of this lesson, we have studied about plasma. What is a plasma? See, when you remove the blood cells, say for example, red blood cells, white blood cells, all, when you remove from the blood, the remaining fluid is called plasma. Okay, blood gets its color because of the RBC cells, no? See, when you remove the RBC cells, blood becomes colorless. So you have studied already that the color of the plasma is straw colored. It loses its color when the red blood cells, which gives red color to the blood, is removed. Clear? So this plasma, after the removal of all the cells like red blood cells and white blood cells, have water in them, enzymes in them. Along with that, they also have a structure called as antibodies. So what plasma has? Plasma is the place, is the liquid which can contain a structure called as antibodies. So these are the structures of antibodies. Again, just like the antigens, antibodies are of two types called antibody A and antibody B. What are the two types? Antibody A and antibody B. If you observe carefully, when I have written the name for antigen, I used capital A and B, whereas for antibodies I have used small alphabets A and B. Always represent when you write, because again and again I cannot tell A. When I tell A, you will think that is it for antigen or antibody. So I am just differentiating by writing capital A and B for antigen and small a and b for antibodies. Clear? So antibodies are proteins. They are present in the plasma of the blood and they contain two types of antibodies called antibody A and antibody B. See, this is the structure of antibody A and this is the structure of antibody B. What is the difference? See, initially for the structure of antigens, if you recall, the structure of antigen A was like this and the structure of antigen B was like this, right? So, such a structure was attached to the RBC cells, whereas you can see complementary structure for antibodies. See, this is antibody A, this is antibody A and this is antibody B. See, this is antigen, AG is antigen, okay, the short form for antigen is AG, okay, this is antigen A, this is antigen B, this structure. Now, if you observe carefully, antigen B can fix with antibody A. It will be like lock and key. Exactly, it will have complementary structure so that it can fit. Similarly, this antigen A can fix like a lock and key, it can fix like this, such structure will be. Just keep this in mind, I am going to explain about this in your later slides, clear. So, just know for now that there are proteins present in the plasma and antibodies are of two types called A and B. Okay. First, in the year 1900, okay, in the year 1900, a man called Carl Landsteiner. What's the name of the man? Carl Landsteiner. This man identified that there are four types of blood groups. It is not a single type, but it is of four types called A blood group, B blood group, AB blood group and O blood group. It was Carl Landsteiner who found that blood groups are of AB, AB and O type. Okay. Now we learn in detail about antigens and antibodies. This is a person with blood group A, this is a person with blood group B, this is a person with blood group C and this column is for a person with blood group O. Clear? So, a person with blood group A will have antigens like this, like circles he will have antigens. Okay. So, A antigen is present on the surface of RBC cells. A person with blood group B will have small diamond like structure on the surface. Okay. Now see, a person with AB blood group will have antigen A as well as antigen B. These round structures and diamond like structures, no, they are the antigens present on the surface. Now, a person with AB blood group will have both antigens A as well as antigens B on the surface. That is why they are called as AB blood group people. And if you see O, he will not have any antigens on the surface of the RBC cells. So, remember this picture clearly in your mind so that easily you can remember this concept. So, A with circled antigens, B with diamond antigens, C with both circle and diamond antigens and O with no antigens. This is in the case of antigens in people's blood group. What about antibodies? Similar to the antigens, a person will be having antibodies in the plasma of his blood. Simultaneously, a person will be having antigens on the surface of the RBC cells as well as antibodies in the plasma of his blood. Okay. So, now the antibodies shape we have already studied in the slide where I have displayed for antibodies. You know, this is antibody B and this is antibody A, you know, right? So, now a person with A blood group with A antigen will have B antibodies in his plasma At the same time, a person with B antigens will have A antibodies in the plasma. What about AB? A person with A and B antigens in his on his RBC cells will not have antibodies at all in the plasma, and person with O blood group will not have any antigen but will have A antibodies as well as B antibodies in the blood group. So remember the slide carefully because it's going to help you lifelong. This is a very basic concept yet very important concept in your day-to-day life as well as in competitive exams also you will be getting. So remember this carefully. Okay. Now there is a chart representing donor and recipient. Suppose if a person needs to donate blood, who can donate to who? You should know, right? So a person with O blood group can donate blood to all the groups, to O, to A group people, to B group people and to AB group people. See, this is represented in green color. O blood group is represented in green color. So a person with O blood group can give blood to O blood group person, A blood group person, B blood group person, AB blood group person. A person with A blood group can donate blood for A and AB. A person with B blood group can donate, see it is represented in red color, to B and AB alone, and AB blood group people can donate blood only to AB group people. Okay, why? I will tell you the reason later, but remember this concept: A only to A and AB, B only to B and AB, and AB only to AB, and O they can donate to all type of blood group people. O can donate. Why so? Because there is a concept called universal donors and receptors. Who is a donor? Donation word all you must have heard, no? Donor means giving. The person who gives is donor. Recipient means or receptors means those who receives is called recipient or receptors. Now who gives here? Universal donor, which means some group of blood group people can donate blood to everyone. Who is that? Already in the chart we have studied, no? O blood group people can give blood to everyone and they are called as universal donors. Who is universal donor? O blood group people, because they can donate their blood to any receptors, either A, B, AB and O blood group people they can donate. So they are called as universal donors. Who is a universal recipient? AB blood group people are universal recipients. They can accept any type of blood group, either A, B, O or AB blood group. These universal recipient group, that is AB blood group people can accept. So they are called as they are called as recipient group. So universal donors are O blood group people and universal recipients are AB blood group people. Then the last part of today's class is about transfusion. Have you heard this word, blood transfusion? What does it mean? Transfusion means transfer of blood from a donor to a recipient. Who is a donor? Somebody who gives the blood is called the donor and someone who receives is called the recipient. So transfusion is the transfer of blood from a donor to a recipient. For this there is one condition: the blood group should be compatible. What is this compatibility? Already we have studied a chart, no? Which blood group person can donate blood to which group of people we have already studied, no? Only that will be compatible. All blood groups cannot be given to one another. Only according to that rule the blood can be transfused or it can be transferred. Okay, when is blood transfusion necessary? Under what conditions blood is transfused? This is especially during accidents, during emergency conditions all, a person loses blood and he may face death if blood is not transfused, no. So in that conditions a blood is transfused. And also if a person is anemic, who is anemic patient, person with low RBC count is considered to be anemic. To that people also blood is transferred. And also to cancer patients also blood is transferred. Okay, now we have learnt the set of rules, no, which blood group people should transfer blood to which blood group people, we have learnt already. Suppose what will happen if we break that rule? If there is mismatch in the blood group, what will happen? What will happen? Why it is given as a rule that only particular group of people should transfer blood to particular blood group? If that rule is not followed, the blood will clump, the antigen and antibody will react and it will form, the blood is usually fluid in nature, no. If there is a clumping of blood because of mismatch in the blood group, the clump will block the blood vessels, it will clog, which means it will block the blood vessels and the blood will not be a fluid anymore, it will become like a clot. See, if you get a cut in your hand all, when blood comes out after some time, it clots, no. Similarly, it will clot inside the blood vessels so that it cannot flow further and the person will face death. So, such dangerous it is when you transfuse blood groups of mismatch type. Clear? So, what we have studied about transfusion? Transfusion is the transfer of blood from a donor to a recipient, provided we have to follow the rule from which blood group people to which blood group people it should be transferred. We have to follow the rule of compatibility. In the case of emergencies like accidents or anemic patients or cancer patients, blood is frequently transferred, and then if at all there is going to be mismatch in the blood, it will lead to clumping of the blood, which means it becomes a solid instead of a liquid, so it cannot flow further in the capillaries, which will lead to death. This is about blood transfusion. For homework, in your next zoology class, you will have a online test in circulatory system. Read from pages 84 to 95 and also learn the worksheet answers also and get prepared for the test. It will be on 23/11/2020. Okay, that is all for today's class, and until I meet you in one more class, you all take care and bye bye.